Here we go. It's day 3 of 4. Remember, day's 4 session. I think the time, my goal is by Saturday at noon. You're going to be able to find that up there, which takes much longer than you think it does, but it will be up there by noon on Saturday. So here we go. Let's go ahead and start on where we left off yesterday, cellular energy. See if you can help me out while you can. ATP versus ADP, you should know the differences. Who thinks they can summarize it real quick? Go ahead.
ATP has 3 phosphates, but ADP only has 2.
You're a genius, yeah. ADP and ATP, they're basically the same molecule. The main difference is one of them has 3 phosphates. 1, 2, 3. And the other one only has 2 phosphates. 1, 2. The one with 3, the triphosphate molecule, is a high energy molecule, whereas ADP, the one with only 2, is a low energy molecule. They actually form a relationship that's cyclic, or cyclic. I've never learned how to say that word properly. ADP and ATP form a cycle, so if you basically break off a phosphate or you add a phosphate, you can change which molecule they are. Anytime we remove phosphates from that molecule, we can release a lot of energy, which in turn means that we can actually do a lot of cool stuff, a lot of cool features inside of our bodies. 38 says photosynthesis, the process of converting light energy to chemical energy is what that should say. Sorry. Chemical energy is something called sugar. We convert solar energy into sugar during the process of photosynthesis. Not us, but plants do. So number 38 goes on and it says, where does it occur? So where does photosynthesis happen? Yes, sir. You're right. It happens in the chloroplast. I'm also going to give you a little hint here. Chloroplasts are mostly found in the leaves of plants. The big wide green things, those are green because they have chlorophyll, which is the energy absorbing molecule of a chloroplast. We're not going to get too much into the structure of the chloroplast, but just to say it, there's a thing called a thylakoid, which is in a stack called a grana, and then we have the space in between those stacks. Those are called the stroma, and that's where all photosynthesis happens, inside the thylakoid. Anyway, so once we have the location done, we're talking about the amount of energy produced, or what kind of energy is produced. Well, we don't really measure it in ATP because it doesn't make a whole lot of ATP. What it does is it stores energy in sugar. Once again, it stores energy in sugar. It's not simply making energy, it's putting it somewhere for later. This is because plants also have mitochondria, because they can use that mitochondria to break down this sugar and make their energy later on. We like sugar. We eat plants for their sugar. Other animals eat plants for their sugar, but they have to use a mitochondria to break it down. So do plants. Doing okay there? Okay. And then, how many stages are actually occurring during photosynthesis? I'm sorry. I skipped one, yeah. Chemical formula. You should know this. Like, a tattoo on each arm would be nice here. One for photosynthesis, one for cell respiration. Maybe you'd only need to get one tattoo if you do it right. But chemical formula is going to be carbon dioxide gas and water and sunlight, or light of some kind, react to form sugar and oxygen. I just said out loud what these letters and numbers mean. Remember, this is the formula for sugar. You should definitely be able to tell what oxygen is, what water is, and what CO2 is, and you should know these are the things that go in that proper order. Remember what we discussed yesterday, everything on the left-hand side, what do we call that? Reactant, right? And then everything on the right-hand side, we call that a product. Yes?
You would still be correct in saying that, like, the arrow is, like, the light source?
It's kind of part of that process, yeah. If I'm saying what are the reactants, though, go ahead and kind of include it under that, yeah. Okay, good question. And then part D actually says, how many stages are there? The answer is there are 2 stages. We have light-dependent and light-independent stages. Let me go ahead and break this down for you. We'll do the first one here first. Light-dependent reactions, of course, depend on the sun, or depend on light of some kind. There are inputs and outputs to this. Input leads to an output. So what is brought in during the light-dependent reactions? Well, of course, light, yeah. That's a giveaway. But the other thing that's brought in there is actually water. And the output from this process is going to be oxygen gas. That's the byproduct that's formed right there. That's where it's formed during the first stage of photosynthesis. When we look at light-independent reactions, they sometimes have a cool name. It's called the Calvin cycle. And their input and output is different as well. The input for the Calvin cycle, that is going to be CO2, and what that's going to output is going to be sugar. I'm not going to write out the formula. This actually makes sense when you start to break it down, because if you take in light and water, what we're doing is we're splitting the water into hydrogen and oxygen. Once you do that, we get rid of the oxygen in this stage, so that means hydrogen here is going to be sent to this stage. CO2 is carbon dioxide. CO2 needs H's in it to become sugar. Hey, look, we're sending H's from the input of one and adding it to the Calvin cycle here, which means we can get CH and O, which would make sugar. So that's how sugar's produced. You still got to have the CO2. Literally, that stuff you breathe out becomes sugar later on. How you doing? That's the most concise summary I've ever made for photosynthesis in my entire life. I'm the best. Yes, ma'am.
So what are some of like the similarities and differences between the Krebs and the Calvin cycle? Like
They're both, well, of course, they're both cycles, right? The Krebs cycle is a cycle, the Calvin cycle is a cycle. They use different intermediate molecules, but basically they're kind of a reversed order of each other. They're still basically carbon affixing or carbon breaking. That would be the main distinctions that would, well, actually really similarities between them. Beyond that, you're going to see that there are different molecules involved. You're going to see different amounts of energy being expended in that process. One will create energy. One's going to basically use energy to store energy. So that'd be about their similarities and differences. Okay, can I move on from photosynthesis?
yeah.
Are we good? Okay, so we have aerobic cell respiration. Aerobic cell respiration is when we break down sugar to make ATP in the presence of oxygen, O2. Please do not forget, aerobic means oxygen is there. Anaerobic means, good, no oxygen is involved, right? So we're talking about an aerobic form of cell respiration. And where does that occur? The mitochondria, yeah. So that should be taking place in all kinds of cells all around the place, just because a lot of cells, especially all eukaryotic cells, have mitochondria. That's how they break down they break down sugar to make energy. The energy that's produced, I'm just going to throw this out there, it produces up to 36 ATP. That's actually including the process of glycolysis. Technically, if you wanted to get all picky, we could say it actually produces 32 to 34 ATP. Do you have to know that? No, not necessarily, but hey, it's a thing and I just wrote it. So what's the chemical formula? It's the same thing as photosynthesis, but in reversed order. I should go ahead and say, once again, sugar and oxygen are going to be brought into this situation. And both now sugar and oxygen, since they're on the left-hand side, they are going to be the reactants, right? That means everything on the right-hand side, those are going to be the products. We're going to produce carbon dioxide gas, that stuff you breathe out. You're going to produce water. That's right, you do produce some water when you breathe out. And ATP, that energy that we just talked about, and a good amount of it, I should mention. I should also mention another byproduct would be heat. You know, we keep ourselves warm by using that same heat source. So cell respiration is going to help us do that. Part D says, how many stages are there in cell respiration? I don't want to get too fancy on you, but there's only 2 stages. The Krebs cycle, which was mentioned earlier, it's sometimes called the citric acid cycle, and then there's the electron transport chain. This is associated with some funny words like oxidative phosphorylation and chemoosmosis. For now, you don't have to know those things, but you should know that the electron transport chain, this part right here, it produces a huge most of the ATP. That last part is really important because this molecule ATP synthase allows these hydrogens to move through it, which helps us lock on more phosphates to ADP, which turns them into ATP, which means, hey, we've got a lot more energy that we can use later or elsewhere in the cell. So that would be there. I'm also just going to go ahead and mention this now, even though it'll show up later. Technically, this thing called glycolysis, this takes place before aerobic cell respiration. So it's going to happen before the Krebs cycle. That's a reality. We'll talk about what glycolysis here is more so in a second, and it relates to anaerobic respiration too, because it itself is anaerobic. So it's got to take place before any anaerobic form of respiration or aerobic form of respiration. But there's a note coming up on that. So some people are still writing. I'm going to take a nice sip of cocoa. Ah, that's delicious. I hope everybody listening to that on YouTube gets the sensation of chocolate filling their nostrils.
ASMR.
Yeah.
Oh, look, we're making the real content.
Yeah. Really getting to the good stuff. And mukbang videos, I'll be making those too. You'll just be watching me eat my peanut butter and jelly. All right, moving on. Okay. So, cellular energy, we're still on the topic because we're talking about anaerobic cell respiration, also known as fermentation. This is a non-oxygen using form of energy production, which means it makes a low amount of ATP. Just as a heads up, it makes 2 ATP. And this relates to a lot of forms of respiration, which are anaerobic. We only talked about 2 in particular, I guess 3 if you want to get technical, but here we go. First one, of course, is going to be glycolysis. I just mentioned it on the last slide. It's a process that takes place before any form of respiration. Doesn't matter if it's anaerobic or aerobic, which is what that means right here. It doesn't matter if it's one of these two. It's going to have glycolysis happen first. Also, this happens in an area outside of mitochondria. It does not happen in the mitochondria. It happens outside of it before it ever gets to a mitochondria. And heck, some forms of, actually all other forms of fermentation don't even happen inside of the mitochondria. It all happens wherever glycolysis happens. The reason for that is because fermentation is just basically hacking glycolysis. It's that same process that we're repurposing what its end products are. So we just tweak that a little bit more. Can I show you what they are? Yeah? So we've got lactic acid fermentation. Hopefully that's easy for you to remember what its byproduct is. It says it in the title. Lactic acid fermentation makes lactic acid, sometimes called lactate. So lactic acid fermentation is the stuff that, or process that occurs inside of your muscles whenever you work them out extremely hard, like I did in 9th grade and embarrassed myself because I couldn't wash my greasy hair and I couldn't move my arms for a week. And then we have alcohol fermentation. That also tells you one of the things that are made in this process, but please do not forget this. There is a distinction. Alcohol fermentation makes CO2 gas. Just to kind of draw this out for you, in glycolysis, we go from a sugar to breaking it down into 2 pyruvates. Okay? In alcohol fermentation, we go from sugar to making 2 pyruvates to turning these into 2 alcohols and 2 2 molecules of CO2. This means this byproduct of CO2, this is going to create those bubbles that are found in things like champagne or beer. But also, it's the same thing that creates the bubbles in your pizza dough when you cause it to rise, or your bread when you cause it to rise. Those gas bubbles are being trapped in the gluten, and therefore you bake it and it keeps that shape. Yeah, that means pizza dough would have some alcohol in it while it's being made, but we bake that out because, you know, ovens are really hot and alcohol evaporates pretty quick. Same thing's true with lactic acid fermentation, but instead of pyruvates, we turn those pyruvates into lactic acid, which still has 3 carbons, so it's all good. Yes?
Where it says 2 pyruvates,
Yeah.
they make 2 alcohol and 2 CO2.
Right here, yeah, it makes 2 alcohol, which are only 2 carbon molecules, and then 2 CO2, which are 2 carbon dioxides. Questions? You sure? Okay. Boop. I'm waiting. I'm waiting. I'm taking another sip of the cocoa. Okay. It was delicious anyway. All right. So Plant structures, you're going to see that we're going to go real short through this. The basic structures, if you don't know what these are, you probably didn't pay very much attention in 3rd grade. But leaves, they're used to capture sunlight because photosynthesis happens in the leaves. So you want to have a wide surface area so you can capture as much light as possible. Power up those chlorophyll inside of your chloroplast to make this process work even better. Roots, well, their job is to anchor the plant to the ground so they don't just fall over. That'd be kind of bad if a little breeze came along and just knocked every tree over. They would all die, and we probably would too. And then, of course, they absorb water from the ground and also the nutrients that are associated with it, which is really, really important for photosynthesis to have water, and that's how they get it. And then, of course, we have the stems. These are the things that connect the roots to the leaves. Usually we store a lot of sugars here, but we also transport things. That's why it's filled with the stuff called vascular tissue. We have 2 forms, xylem and phloem. Try to keep in mind xylem, xylem. Remember that goofy thing? Xylem, skylem, so that means it brings water up from the roots towards the sky. Sky, skylem, okay. And then we have phloem. This moves products around. Mostly it moves sugar around in plants. And these are the products of photosynthesis. Sugar is, and also some oxygen is going to be moved around in these things. These are vascular tissue, which means like arteries and veins, but for plants. So this is how they transport all their materials. Doing okay? Yeah? Waiting. Still waiting. I watched an ASMR video yesterday of a man eating 2 Big Macs, and each Big Mac only took 3 bites. I don't remember what it stands for, but it's when like the sound is really good, and like when you hear people chewing and crunching, it kind of comforts people. And for some people, it stimulates this tingly, nice sensation in the back of their head. So it kind of makes their hair stand up, and they enjoy that sensation. So you can hear people like talking very softly. It's kind of odd. I don't know. Anyway, so the sexual organs for plants, these should kind of stand out to you. We have a male and a female version. Does anybody remember what the female or male version is called? Yeah, which one's male? Stamen. It's easy to remember because Ben's in the title. So that tells us carpal is the female reproductive system. I'm just going to tag this in there. The pistil is the exact same thing. It's spelled P-I-S-T-I-L. Funny way to spell it. There it is. It's the female reproductive system. It's made up of 3 components. The first component is going to be the stigma. That's going to be the thing that captures pollen from the environment. I'm going to draw this for you. It's going to look like I'm drawing like a little turkey leg, but it's not a turkey leg. It's supposed to be an actual female portion of a plant. So this top portion right here, this is in fact the stigma. And it's sticky. I always see stigma and stigma, stigma, sticky, sticky, stigma, because their job is to catch pollen. Actually, pollen will stick to it. The pollen contains the sperm, so these will open up and eventually drop the sperm down a tube, and this tube is called the pollen tube, appropriately enough, and it'll travel down this thing called the style. You know, it's kind of the equivalent, yeah. That's actually a really good analogy. It's about the same. Yeah, I couldn't do a better job there. You're right. It would be like the fallopian tube. And also, it's easy to remember because most females have more style than men. And then we have the ovary. Hey, the ovary is definitely a female reproductive organ. You should know that. That's like the primary sexual organ for all females. So, yeah, congratulations. Plants might have more than 2, but hey, whatever. In this case, for the flower, it's usually just 1. So yeah, that's going to supply resources for the future seed. A lot of the times they store a lot of sugar, which helped us remember how gross apples seem to be now, because they're just the swollen ovary of a flower. And you eat swollen ovaries sometimes, which is, you barbarian, you savage. How dare you? Stamen, easy to remember. Once again, men is in the title, so it tells you it's the male reproductive organ. It's only made up of 2 parts, which, by the way, I forgot to draw this here. This would be the ovary down here. There could be like little future seeds down here, and those become fertilized. The stamen, the male reproductive system, it's made of 2 parts, the anther and the filament. The filament is just a long tube, and it just extends away from the body of the flower, but then the anther is filled with little dots that are the actual pollen. So they produce the pollen, in turn they produce the sperm, and they're going to fertilize things in the future. So when it comes to the anther, once again, it's the top. It's supposed to be removed far away from the body because we want a pollinator, or maybe the wind, to come by and move that pollen off and carry it to another flower. The filament's job is to do that very thing, to pull it away from the body of the flower so it's more likely to be taken away. So once again, anther is the top part. Letter I there. And double I there is going to be the filament, that long tube. Yeah?
No.
Okay, nose. Lots of nose.
The pistil and the carpal are like interchangeable words.
Yep, they're interchangeable words. Mean the same thing. I think pistil is a more old-fashioned way. I think carpal is a more current word. But I'm not a botanist, so I don't know. Can I hit the button?
Take a sip. Take a sip. Take a sip.
Take a loud
I'm going to rearrange my papers really briefly.
Yeah. Yeah. We want to hear that.
I'm going to read ahead for a second. I'm reading ahead. Can't see, I'm reading. My eyes are on the paper. What?
Have you seen The Martian?
Yeah?
Is that like possible?
Oh, no, not really. Okay, so the cell cycle, the main stages of the cell cycle, they are going to be G1, S, G2, mitosis, and cytokinesis. Just to point out to you, the main stages are like the life cycle of the cell. I'm going to draw a circle that is going to be basically a pie chart for us. Look at me slowing down, making good circles. Good for me. Good job, Olson. We'll do like this, and we'll do like this. Ooh, good job. So that's going to be G1, synthesis, G2, mitosis, and then I'm going to make this line extra thick because it's going to represent this little thing for us. So the first thing that we're going to have involved in the life of a cell is they're going to go through a stage called G1, which stands for gap 1, which means growth. Every time you see G in this phrasing here, it should tell you that the cell is probably growing. It does some minor checks. As a matter of fact, right here at the end of gap 1, I'm going to put a little square there. At the end of gap 1, there is a check to make sure the cell is healthy enough or ready enough to go through synthesis stage. Because what occurs during synthesis stage? The DNA is going to get copied or replicated or duplicated. Yes, sir.
Or if it's not healthy enough.
If it's not healthy enough, a lot of the times the cell will go, hey, you're not healthy enough. Go away, and it'll usually self-destruct and usually get recycled and get used to make other cells in the future. If it's a bad cell, we don't want it to reproduce because that could lead to some other problems. After synthesis, we move on to gap 2. Gap 2 is also growing because gap should mean growth for you. And it checks for mistakes before we actually divide the cell. I will draw another box up here for you. It's another space for the cell to stop and check and see if it's prepared to actually rip itself in half. This is really important because if there's a mistake here, we can have problems with chromosomes, we can have genetic deformations, and it can even lead to things like cancer. We now see in M phase, or mitosis phase, the dividing of the cell's nucleus. Please don't forget, this just says cell nucleus. Mitosis is not a division of the cell, it's a division of the cell's nucleus, the nuclear material in it as well. Everything else beyond the nucleus, that's going to be followed up with the process of cytokinesis. That's when we divide the cell's cytoplasm and we end up with 2 separate living cells that are different than the host. Remember, I made you answer some pretty tricky questions previously. The cell doesn't really die, okay? The cell just becomes parts of future cells. So in a way, you're kind of like pressing things forward and you're always kind of moving forward. And that's kind of nice. But at this point, there's no original parts left, so you've been replaced. What's that?
I said unless they kill themselves.
I guess sometimes they could. So we have those things written down. I want to go ahead and just mention this because kids were asking me about it in another class period. Yes, you can have some cases where you have a cell that's stuck in gap 0, meaning the cell is stuck in senescence. The cell is like kind of asleep. It doesn't go through division. It doesn't reproduce. It doesn't change. It wants to stay in that state. Yes?
Isn't that brain cells?
Brain cells and liver cells are notoriously stuck in the stage. Liver cells can kick themselves out of it and move on and grow. It just takes a long time. Brain cells, they don't do that. We'd have to do something very special to make them do that. In a lot of cases means probably using something like a stem cell to make that happen. But we'll talk about that more later on next year, which is funny to say because that's a year. That's like next year, 2018. Yeah, I think that's pretty good. I think we're doing fine. Do you guys have any questions on the stages of the cell cycle? Oh, can I just mention one thing? Sorry, I should have mentioned this earlier. I didn't make a note of this and other things. These three things together, they have a name. It's called interphase. You should definitely know that gap 1, synthesis, and gap 2 all together are referred to as interphase, and they make up like 95% of a cell's life. So most of the time, the cell is going to be stuck in interphase. And most of the time, it's going to function normally as well. Yeah?
Is that gap 1 and
Gap 1, synthesis, and gap 2. I tried to highlight them here for you. So it'd be gap 1, synthesis, gap 2. These together are collectively known as interphase. So whenever we see that listed in like stages of, the cell cycle, we might just reference it as interphase because it includes all three of these. Any questions? Doing good?
Yeah.
All right. So when it comes to mitosis, we're talking about these different individual stages that we find involved. First off, we have something called prophase. I'll try to draw them out for you in a simple way, just to make it easy. So here comes some drawings. Get ready. That's okay. That's a circle still. Ooh, that's an even better circle. We'll make this one a little bit wider. Good. And then a peanut shape. Good. Nice, Olson. So prophase, this one right here, I always keep writing my P's as a D. Prophase is going to include the chromatin condensing. So instead of like a loose ball of yarn, we end up with just like little X's inside of what used to be the nucleus, but it's dissolved. And it's allowing these, it's going to allow these chromosomes to move in different directions. We also have these things called centromeres. They're going to start growing, these little spindle fibers, and that's going to start moving to the poles of the cell. This all technically happens during something called prometaphase, where things just move a lot, but then that jumps to the next stage called metaphase, but I want to talk about something else first. Yeah, they are. The spindle fibers are all protein structures, and they'd be associated with the cytoskeleton. I just want to mention really quick a chromosome. I know we've been using that word a lot lately. Don't forget that a chromosome kind of looks like this in most cases, right? That entire thing is a chromosome, but if I just look at one side of that, we refer to this as a chromatid. Okay, and there's even this little belt that connects these two halves together. This is referred to as a centromere. Okay, so we're going to move these things apart by using a centromere. So in the next stage, in metaphase, these chromosomes are going to align in the middle of the cell. These long spindle fibers are now going to attach to the middle of these centromeres, and they're going to rip them apart. Remember the giant spiders that rip in half the children? Yeah, that's what we're saying. The chromosomes are attached on two sides. And then we're going to like basically pull and rip those things in half. That's what we call anaphase. So these centrioles are now going to start shortening those spindle fibers, and they're going to start moving to opposite sides of the cell. So it'd look kind of like this. You'll see that the cell will start to divide right through the middle. Once again, in metaphase, that does form something called a metaphase plate. I'm only bringing that up because in your cell cycle web quests I made you guys do, that actually was one of those explicit things that were in there. You need to know about that metaphase plate. That's the distinguishing characteristic of what makes metaphase, is that they form a straight line at the equator of the cell. And then we have telophase. In telophase, the nuclei start to reform. I don't know if nuclei is a word, but I made it a word. And then we have those chromosomes starting to loosen back up. They don't take a particular shape again. They'll start to eventually reform chromatin. You can start to see that the cell is going to start to divide. And that splitting, we actually call that a cleavage furrow. In plant cells, we call this a cell plate. And that cell plate is going to be basically a reforming of the cell wall. Because remember, plant cells have cell walls. After this, we end up actually dividing the cell, and that would be cytokinesis. But this tells us that there's only one stage of division, okay? Only one stage of division is found in mitosis. I should also mention that the products of mitosis make two genetically identical diploid cells. Diploid means it's got two copies of its genetic information. So when we're saying in mitosis, we start off with one diploid cell. We are making two diploid cells. They are exactly the same to each other, which actually tells us, since they're identical cells, they're the same. This is used in what we call body cells, or you might know them as somatic cells. Same thing. Somatic cells, body cells. Somatic's just a funny, cool, awesome, sciencey way of saying body cells. So those are all your cells in your body except for your sex cells or your gametes, which would be involved in meiosis. Do you guys have any questions there? I think we're making great time. I think we can do it. We can pull through, guys.
What time do we get up?
28 or 38 or whatever it is. Yeah, 28. So we have 8 minutes. 2 more topics. Let's do this thing. You ready? Final push.
Let's go.
Okay. Meiosis. Meiosis means it's got stages of division. Meiosis is one form of cell division that creates gametes or sex cells. Remember, both terms are interchangeable, but sex cells, the good examples of those would be egg and sperm. Remember, the egg is the female reproductive sex cell, and the sperm is the male reproductive sex cell. Each of them only have half the genetic information from each parent, which means if an egg meets a sperm, there's 2 halves that are forming together to make a whole, which makes a new diploid cell. But that only happens during fertilization, not during meiosis itself. So we have those actual stages of division. There are 2 stages of division, which we refer to as meiosis 1 and meiosis 2. I misspelled meiosis right here, so hopefully I'll fix that before I post it online. Let me write this out for you. We've got interphase, prophase, metaphase, anaphase, telophase, just like mitosis, but now we have cytokinesis, and then back to prophase, metaphase, anaphase, telophase, and cytokinesis. It gets kind of confusing if we don't explicitly state like which form of prophase we're talking about or anaphase we're talking about. So we put a little 1 below the first half here and a little 2 below the second half here. That tells us that it's either meiosis 1 or meiosis 2. So meiosis 1 happens first, meiosis 2 happens second. Meiosis 2 is more like mitosis because it just splits things, but some weird stuff happens in meiosis 1. So the products of meiosis, there are 4 genetically unique daughter cells, which are actually called haploid cells. So we go from 1 diploid cell, just 1, and we produce 4 haploid cells. Doing okay? No questions there? Remember, haploid means it has half the genetic information. And once again, that's why 2 sex cells have to come together to form a new diploid cell, a new zygote, which is what you were when you were first made. C says crossing over. Crossing over is a process that increases genetic diversity by having homologous chromosomes swap small sections of genes or genetic material. I should mention that this happens during prophase 1. Okay, so it happens during meiosis 1. You guys might remember this, but our chromosomes, I'm going to try to draw this as best as I can, as quick as I can. Our chromosomes like to hug each other. These same sized chromosomes, they like to hug each other. And since they're so close to each other, what they end up doing is exchanging some genetic material. It's actually random what materials is exchanged, so that actually makes everything kind of more unique. So the red gets a little blue in it, the blue gets a little red in it, and that's the crossing over process. Then we have independent assortment. This is a tricky topic for a lot of kids to remember, but it relates to genetics. It talks about why we set up Punnett squares the way we do. We're saying we increase genetic diversity because each division of the chromosomes doesn't depend on how the other chromosomes were divided. We studied this in 2 ways. If you guys remember the Dragon Genetics Lab, we had those popsicle sticks. Those aren't the letter J, those are popsicle sticks. One was orange, one was red, one was yellow, one was green. We even had a sex chromosome. But the point is, the green chromosome, whatever letters were on the green chromosome, didn't affect what letters were on the orange chromosome, or what were on the on the yellow chromosome, or the orange chromosome, or the red chromosome. They're all separate from each other, and it didn't influence one way or the other what happened on the green to the other colors. That is independent assortment. Having a harder time understanding that? Check this out. We have homologous chromosomes.
Does that also happen in prophase 1?
Oh, no, that actually happens during metaphase 1. Good question. During metaphase 1. I guess technically anaphase 1, but that's fine. So anyway, we could have chromosomes that line up in different ways. For instance, in this one, they're going to split down the middle here. So once the cell splits in 2 different directions, we'll get all red on this side and blue on another side. These things could split themselves so that we end up with a big red over here and a little red over here, which means for our blue side, we'd end up with a little blue, a big blue, a big blue, and a little blue. The randomness of how they arrange themselves, it doesn't matter. They can arrange themselves however they want. We can see different possibilities form. Okay? Moving on real quick. Okay, cancer. Cancer is basically when no proper checks in the cell cycle occur. We usually refer to these as unregulated cell growth, which is what number 47 says right there. It's basically a mistake in the cells replicated, and it's allowed to make more cells, which keep building up on themselves, forming like a tumor, and which could eventually be dangerous because they could spread. There can be different causes to cancer, like carcinogens. Sometimes you have a genetic predisposition, which doesn't guarantee cancer, just means you're more likely to get it. And of course, there are other factors too, like radiation, which I guess is a carcinogen too. I want to go ahead and mention binary fission right here. This is when simple cells like bacteria or prokaryotes, when they reproduce themselves, what they're doing is they're trying to divide, but they don't use precise stages. The reason for that is because, well, they don't have a nucleus. So how could they go through mitosis, the division of the cell's nucleus, if they don't have a nucleus? So they don't. They just copy their DNA and rip it in half and hope for the best. They split the cell and just hope 2 halves of DNA end up on both sides, which means they can reproduce really, 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 really fast.